Hello everyone. Today we are going to be talking about 7B muscle movement and the heart from the IAL Edexcel Biology Unit 5. So let us begin. Okay, so firstly, let's look at how muscles, tendons, the skeleton and ligaments interact to enable movement, including antagonistic muscle pairs, extensors and flexors. So let's look at the figure shown in the slide here. This red line is the tendon which joins muscles to bones, and then this is a bone. And this yellow line that we see here, a small, narrow sort of line, it's called the cartilage. This absorbs the synovial fluid, which and acts as a shock absorber. The red part portion, the meaty part, is the muscle, and the narrow red part is the ligament which joins bone to bone, and it is very strong and flexible. The synovial fluid is the blue part here, and the synovial membrane is the bluish part over here. The, the white part, the very narrow white part, is the fibrous capsule which encloses the joint, and there is the last structure that is the pad of cartilage, which gives additional protection. How does muscle bring about movement? Muscles bring about movement at a joint. Most movements are produced by the coordinated action of several muscles. So muscles shorten, pulling on the bone and smoothing the joint. Muscle can only pull, but they cannot push. A pair of muscles that work in this way are described as antagonistic. So, antagonistic basically means that a pair of muscles that can contract, another pair will relax. So that's how a muscle movement is coordinated. For example, when you flex your knee by contracting the hamstring muscles at the back of the thigh, the quads at the front of the thigh relax and so are stretched. To extend the knee, the quadriceps shorten while the hamstrings relax. A muscle that contracts to cause extension of joint is called an extensor, and the corresponding flexor muscle, which contracts, is to reverse the movement. In the next part, we look at the structural and physiological differences between fast and slow twitch muscle fibers. So basically, here we have categorized the muscle fibers into two categories, which is the fast twitch muscles and the second is the slow twitch muscle fibers. So, what is the difference between the fast twitch and slow twitch muscle fibers? The fast twitch muscles have less mitochondria, while slow twitch muscles have more number of mitochondria, which actually means that fast twitch muscles have a low requirement of oxygen, while slow twitch muscles have a higher requirement of oxygen. So by this we can also find out that since there is a low supply or low requirement of oxygen for fast twitch muscles, so there is more anaerobic respiration is happening, while slow twitch muscles basically require a high level of or high supply of oxygen, has more aerobic respiration. Which brings us to the next point, fast twitch muscles fatigue very rapidly due to lactate buildup, while slow twitch muscles fatigue very slowly due to reduced lactate. Fast twitch muscles have fewer capillaries, while slow twitch muscles have a dense network of capillaries. So fewer capillary means less supply of blood or less blood flow, while slow twitch muscles mean more blood flow. Fast-twitch muscles have a large amount of glycogen stored present, while slow-twitch muscles have small amounts of glycogen stored present. Fast-twitch muscles have a large store of calcium ions in the sarcoplasmic reticulum, and slow-twitch muscles have small store of calcium ions in the sarcoplasmic reticulum. Okay. Next point is faster rate of hydrolysis in the myosin head for fast-twitch muscle, and the slow-twitch muscles have slower rate of hydrolysis in the myosin head. And lastly, slow-twitch muscle is reddish in color because of more blood flow. Fast-twitch muscle is whitish in color due to less blood. Now, one more extra point I would like to add in here is low-twitch muscles have more number of myoglobin, while fast-twitch muscles have less number of myoglobin. Myoglobin is basically sort of an oxygen carrier. So since low-twitch muscles have requirement of higher oxygen, so they have more number of myoglobin, while fast-twitch muscles, which have a lower requirement of oxygen, has less number of myoglobin or little to no myoglobin. Now, please look at the structure of the mammalian skeletal muscle fiber, and this is a very important structure that will come to the exams. It could come as a labeling or just to explain what is the function of each of these structures. Okay, so now we are going to look at what happens to the microstructure during muscle contraction. The sarcomere shortens, which causes this Z disc right over here to move together as in to contract, and the M line remains unchanged, while the I band here shortens and the A band length remains constant. The H zone over here decreases in size. So let's go to a short summary. So as the shortening of sarcomeres is occurring, overall during contraction, the sarcomere shortens that brings the Z disc closer together, decreasing the I band and the H zone, while the A band that remain unchanged. These changes result from the sliding filament mechanism, which we will learn on the next slide, where myosin heads pull actin filaments towards the center of the sarcomere.
So now let's move on to the process of contraction of skeletal muscle in terms of sliding filament theory. So in this process, a nerve impulse arrives at the neuromuscular juncture as the receiver of a stimulus from the sensory neurons. The calcium ions that are stored in the sarcoplasmic reticulum attaches to the troponin molecule, which is a protein, and causes it to move and change shape. As a result, the tropomyosin on the actin filament that is attached to the troponin molecule, which is also a protein, shifts its position and exposes the myosin binding sites on the actin filament. The myosin heads then bind to the exposed myosin binding site on the actin filament, forming a cross bridge known as the actomyosin cross bridge. So when the myosin head binds to the actin, ADP and PI on the myosin head are released. Then the myosin changes shape, causing the myosin head to move not forward. This movement results in the relative movement of the filaments. The attached actin moves over the myosin. An ATP molecule binds to the myosin head after this movement, and this causes the myosin head to detach from the exposed myosin binding site, and which breaks down the ectomyosin bridge. Then ATPase on the myosin head hydrolyzes the ATP. This forms the ADP and PI molecules again, and this hydrolysis causes a change in the shape of the myosin head, and then this myosin head returns to its upright original position. This entire process results in the muscle contraction. Now, if this explanation you find it to be difficult, I want you to look at the figure 7.19, the sliding theory of muscle contraction, and I need you to write down this point and preferably memorize it or understand this really well. This question can come to your exams, and it's a very important question.
Okay, before we move on to the next slide of this chapter, I just quickly wanted to review action of the heart, the cardiac cycle, MAS. Okay, so before we start the actual cardiac cycle, I just wanted to let you know the basics. So, blood from the body, deoxygenated blood from the body, from the arms, the legs, the head, everywhere, enters the right atrium via the inferior and superior vena cava. Right atrium fills with this blood, and as it's filling, the pressure inside the atrium has risen to a higher level, and then this pressure triggers the tricuspid valve that is located in between the right atrium and the right ventricle to open, letting the blood into the right ventricle. This is the atrial contraction. The atrial contracts and then pushes more blood into the ventricle. There is a valve that separates the right atrium and ventricle that ensures blood flows in one direction only, and it is supported by tendinous cords. Okay, so the right ventricle contracts, and this pushes the blood through the pulmonary arteries to the lungs for oxygenation. Now, let's come to the mechanism of the oxygenated blood. Blood returns to the left atrium after getting oxygenated from the lungs through the pulmonary veins, and the left atrium is filled with the blood, oxygenated blood, and then this blood then flows to the left ventricle and the bicuspid valve opens. Now the bicuspid valves are located between the left atrium and the left ventricle, so kind of like same mechanism that happened in the left ventricle or left side of the heart and the right side of the heart. So the left ventricle then contracts and pumps blood all over the body via the aorta. Now, a small thing here, which could be just for a review, not related much, but this side, the right side of the heart, has more pressure than the left side because this side needs to pump blood all over the body, and the right side just needs to pump the designated blood to the lungs. Alright, coming to the next part, our heart is continually contracting and then relaxing. This contraction and relaxation. The contraction of the heart is called the systole, and the systole can be divided into atrial systole when the atria contract together and forces the blood into the ventricles, and when the ventricle and ventricular systole, when the ventricle contract, ventricle systole happened about 0.13 seconds after atrial systole, and we will learn more about this in the next slide. And forces blood out of the ventricle into the pulmonary artery and the aorta. Between contractions, the heart relaxes and this relaxation stage is called diastole. And one cycle of systole and diastole make up a single heartbeat which lasts for about 0.8 seconds in humans. And this is the entire review of the cardiac cycle. We are now prepared to move to the next slide.
See, here is a graph of the entire cardiac cycle. Moving on to the next slide, that is how the normal electrical activity of the heart coordinates the heartbeat, including the roles of the sinoatrial node, the atrioventricular node, and the bundle of His and the Purkin fibers. So, in the sinoatrial node, this, which is located in the right atrium, it basically generates an electrical impulse. That electrical impulse is the wave of depolarization that we will learn more about in topic 8. It initiates the heartbeat. It is often referred to as the pacemaker of the heart because the sinoatrial node sets the rhythm for the whole heart. So basically, we could say that the entire cardiac cycle foundation is laid out by the sinoatrial node because of the sinoatrial node generating an electrical impulse. So this electrical impulse then causes the atria to contract. It is atrial systole. This pushes blood into the ventricle. Now the function of atrioventricular node. The wave of depolarization spreads through the atria and reaches the atrioventricular node. The atrioventricular node is located between the atria and the ventricles. So there is a wall between the atria and ventricle. So there is atrioventricular node located. There is a slight delay at the AVN to ensure that the atria have completely contracted. So we see in the graph, previous slide, that there is a very little time difference between the 0.1 to 0.2, the atrial and ventricular systole, it is the S. So that is because there is this slight delay at the AVN. So this is made sure that the atria have completely contracted. The ventricles have fully filled with blood before the ventricles can contract as well. This delay is really essential for the efficient pumping of the blood. Bundle of His and Purkinje fibers. So after this delay, the impulse is then passed down the bundle of His, which is a group of specialized heart muscle cells. This conduct the electrical signal down the septum. Now, remember what the septum is. The septum is the wall between two ventricles that separate the left side of the heart and the right side of the heart. The bundle of His also further down branches into a Purkinje fiber, which spread the impulses through the walls of these ventricles and causes them to contract from the bottom up. This is called the ventricular systole. This also ensures that blood is efficiently pumped out of the heart and to the lungs via the pulmonary artery and to the rest of the body via the aorta. Now keep in mind that this action is occurring in both the left side of the heart and the right side of the heart. Carrying on with the next slide, how the use of electrocardiograms can aid in the diagnosis of abnormal heart rhythms. So, heart rhythms are not always rhythmic, they can be abnormal. And ECGs are the way, or electrocardiograms are the way to detect these abnormal heart rhythms. Let's look at how we can detect this. The electrical activity of the heart can be detected and displayed on an electrocardiogram. This is a graph, and it's a record of the electrical activity of an individual during their cardiac cycle. This is the most common test to check the problems with heart. The test is relatively easy to carry out, and any patient with suspected cardiac problems will have one. So, in an ECG, firstly, an electrode is attached to the person's chest and limbs to record the electrical currents that is produced during the cardiac cycle. When there is a change in polarization of the cardiac muscle, a small electrical current can be detected at the skin's surface. This is what an ECG may measure. This is what an ECG measures. An ECG is usually performed while the patient is at rest, lying down and relaxed, but it can also be used in an stress test. This involves doing an ECG before and during and after the period of exercise and is used to detect heart problems that emerge only when the heart is working hard. Breathing rate and blood pressure may also be measured and is recorded. Okay, so this one slide is very important and mathematical questions can come from this slide in your exams. So here, please observe the graph in figure 7.44. What does the ECG trace show us? Note which wave on the ECG represents each stage in the electrical activity of the graph. You have to memorize which wave on the ECG represents which stage. Here, from starting from the P wave. P wave here represents the depolarization of the atria and leading to the atrial contraction, atrial system. That is the peak performance. PR interval from the graph represent the time taken for impulses to be conducted from the SN across the atria to the ventricles through the atrioventricular node. So that is the slight delay, that is the PR interval that is taken, the time taken from the synatrial node to the ventricles through the atria. Now the QRS complex represents the wave of depolarization resulting in a contraction of the ventricles, the ventricular systole. That's the QRS complex representing that. The T wave here represents the repolarization recovery of the ventricles during the heart's relaxation phase. So that's diastole. So here T wave is basically representing that after the ventricular systole has occurred, so now the heart, we can say, is the ventricles have been in a relaxation state after the blood has been completely pumped out of the ventricles, and this phase is called diastole. So the P wave represents the diastole. Now, here we also see that the graph is really rhythmic over here, and it has 1, 2, 3 peaks in total. This is a normal functioning heart rhythm, of course, but what if the heart rate of the individual increases? So then the peaks over here will increase because your contraction of the ventricles and atria is occurring more frequently, hence this peak will increase. While what if the heartbeat is slower? Then in that case, the heartbeat right over here, the peaks would decrease due to less contraction, or the peaks would be really slow, really this narrow, sort of, not really sharp. And furthermore, I would like to include that this graph can also hold really mathematical questions, as in calculative questions. So here I have put in a little information about the mathematical part. Please do give it a thorough read and then try to practice. Carrying on to the next slide, define and calculate cardiac output. Variations in ventilation and cardiac output enable rapid delivery of oxygen to tissues and the removal of carbon dioxide from them. So, what is the definition of ventilation? Ventilation refers to the process of air moving in and out of the lungs. It directly affects the amount of oxygen available for gas exchange and the removal of carbon dioxide. So, ventilation, for example, increased ventilation So during activities like exercise, our ventilation rate increases. More increase in ventilation means more oxygen is breathing in. So this helps saturate the blood with more oxygen more quickly. So therefore, we have an enhanced respiratory rate and depth. It is also called tidal volume, which allows for more efficient gas exchange in the alveoli. So here's a quick little definition of the tidal volume. So the tidal volume is basically the volume of air that enters and leaves the lungs at each natural resting breath. Okay. The next part is gas exchange efficiency. Increased ventilation also improves the partial pressure gradient for oxygen and carbon dioxide between the alveoli and blood, facilitating faster diffusion. This also helps remove excess CO2 from the blood, preventing respiratory acidosis. So, you all know if there is an increase in the carbon dioxide level in blood, the pH level falls, so that makes the blood more acidic. So this prevents the respiratory acidosis. In the cardiac output is the volume of blood the heart pumps per minute. It is determined by heart rate and stroke volume, and it is calculated by multiplying cardiac volume by heart rate. In an increased cardiac output during physical activity, the body increases CO to deliver more oxygen rich blood to active tissues. This is achieved by increasing heart rate or stroke volume. Higher carbon, uh, higher CO enhances perfusion of tissues, ensuring they receive adequate oxygen for metabolic processes. So, what is oxygen transport? So, blood is obviously the medium for transporting oxygen from the lungs to the tissues, and with higher CO, the delivery of oxygen to cells increases, matching their metabolic demands. Removal of carbon dioxide. Increased blood flow helps transport CO2, byproduct of cellular metabolism, from tissues to the lungs for exhalation. Efficient removal of CO2 prevents accumulation, which is vital for maintaining acid-base balance in the body. Now, a little previous chapter review. I believe cellular metabolism is basically a cellular respiration, which produces the byproduct carbon dioxide. Now, an important part of the slide, how heart rate and ventilation rate are controlled, and the roles of cardiovascular control center and ventilation center in the medulla oblongata is responsible. So now, sometimes we need to increase the heart rate in order to meet our body's metabolic demand. So, for example, when we are exercising, our body requires much energy for larger ATP production, and so ATP synthesis, as we all know, requires more oxygen level. So we need to increase the heart rate in order to get more oxygen for the body, so that more oxygen can be pumped throughout the entire body. So to increase the heart rate, the stretch pressure baroreceptors, the stretch receptors or pressure receptors detects a change in the environment. Now you will maybe thinking that where is this stretch pressure receptors? So these stretch pressure receptors are located in the carotid artery. So it responds by sending impulses to the heart along the sympathetic nerve, which we will read more about in topic 8, from the cardiovascular center in the medulla oblongata. It is a part of the brain. After the impulse has reached the heart, noradrenaline noradrenaline is released to stimulate the sinoatrial node. So the impulse um, is sent from the cardiovascular center in the medulla oblongata via the sympathetic nerve to the sinoatrial node, and then noradrenaline has been released. So this, we all know that an adrenaline increases heart rate. How? This increases the frequency of signals that is sent from the SAN region to allow the heart to beat more quickly, as well as the branches of this sympathetic nerve pass into the ventricles, so they increase the force of contraction. So we all know that the left ventricle pumps the blood from the aorta to the entire body or different part of the body. So this sympathetic nerves also branches into the ventricles, and then they send signals to the ventricle to increase the force of contraction so that more blood is pumped, more oxygenated blood is pumped out of the ventricles and into the body. Now, the opposite reaction to decrease the heart rate. So the stretch pressure receptors in the carotid artery detects the change in the environment. Firstly, the same mechanism. Then it responds by sending impulses to the heart, but this time along the parasympathetic nerve from the cardiovascular center in the medulla oblongata. And after the impulse has reached the heart, there is a chemical that is released, which is known as acetylcholine, and this inhibits the uh, sinoatrial nodes from increasing the frequency of signals. So this decreases the frequency of signals from the SAN region to allow the heart to beat more slowly. Hence, less amount of blood is pumped to the body. So after this example can be ideally suited in a situation like after the exercise, our heart rate has during the exercise our heart rate has increased. But what happens afterwards? Our heart rate needs to be back in rhythm again. So our body then uses this mechanism to gradually decrease the heart rate back to normal. So the next part of the slide, the role of chemoreceptors in the aorta in reducing the level of carbon dioxide. So as you know that the walls of aorta and carotid arteries contain chemoreceptors and baroreceptors, which are very sensitive to the levels of carbon dioxide in the body. So as the carbon dioxide levels go up, the pH level in the blood decreases, leading to a more acidic sort of environment in the blood. So this decrease in the pH is detected by the receptors, and then they send impulses along the sensory neurons to the cardiovascular control center located in the medulla oblongata. Then this increases the number of impulses that is sent from the cardiovascular control center traveling down the sympathetic nerves to the heart. As a result of this increased nerve impulses, this increases the heart rate and the blood flow to the lungs so that more amount of carbon dioxide is removed from the blood. So as the blood containing more carbon dioxide level falls, the pH rises and returns back to its original natural pH. And then as the pH of the blood increases, returning back to the original pH, the chemoreceptor responds to this change by reducing the impulses sent to the cardiovascular center. As a result, the cardiovascular center then reduces the number of impulses that were sent along the sympathetic nerve to the heart. This can decrease the acceleration of the heart rate, so it returns to the intrinsic rhythm or the original heart rhythm. So let's move on to another slide where very common mathematical questions can come from. So that is spirometer. What is spirometer? Spirometer is basically an instrument to measure the breathing rate or ventilation rate in a person. So before we proceed on actually interpreting or dissecting this graph, let's look at some important definition related to this graph. What is the tidal volume? As I have mentioned in one of the previous slides, tidal volume is the volume of air that enters and leaves the lungs at each natural resting breath. So right now, as I'm sitting here and making this video, my volume of air that I'm breathing in, so the ventilation rate is really natural. I'm not I'm not really quickly breathing, or have not been breathing rapidly. So that means the breath that I'm taking right this moment is my tidal volume. That is the natural resting breath. Next, inspiratory reserve volume. So inspiratory reserve volume is the volume of air that you can take in above the normal inspired tidal volume. So that is basically more than our natural resting breath. In other words, this is the extra air that you can take in when you breathe in as deeply as possible after a normal inspiration. So anytime we take a deep breath, that is, we are taking in extra volume of air, and this is known as the inspiratory reserve volume. What is the expiratory reserve volume? It's quite the opposite of the previous one. Is the volume of air that you can force out above the normal expired tidal volume. So if you're breathing in at a natural resting breath, so that means we are also breathing out at a natural resting breath. So expiratory reserve volume is the volume of air that we breathe out more than the natural breath, exhaling breath out. So this is the extra air you breathe out when you force the air out of your lungs as hard as possible after a normal expiration. What is the vital capacity? The vital capacity is the total of the tidal volume and the inspiratory and expiratory reserves. It is the volume of air which you can breathe out by breathing out as hard as you can and then breathing as deeply as possible. So this is, in basic terms, is the total of the tidal volume and the inspiratory and expiratory reserves. Three things: total of the tidal volume, total of the inspiratory and expiratory reserves. What is the residual volume? Residual volume is the volume of air left in the lungs after the strongest possible expiration. What is the total lung capacity? It is the sum of the vital capacity and the residual volume. And lastly, inspiratory capacity is the volume that can be inspired from the end of a normal expiration. Notice there is a small equation at the bottom and at the end of this definition, which is inspiratory capacity equals to tidal volume plus inspiratory reserve volume. Now, let's come here and actually interpret this graph right over here. So look at figure A. This diagram is based on spirometer trace, gives you an idea of the average lung volume manifest. However, values between men and women and athletes and non-athletes can vary considerably, of course. Okay. In a normal person at rest, tidal volume is about 0.5 decimeter cube, or about 15% of the vital capacity of the lungs. The rate of breathing can be expressed as the ventilation rate. This is a measure of the volume of air breathed into a minute. Okay. So the equation for the ventilation rate is tidal volume into frequency of inspiration. The ventilation of the lungs needs to supply all the oxygen that is required by the tissues of the body whatever they're doing, and also remove all the waste carbon dioxide. The ventilation rate is affected by two things: the amount of air you take into your lungs at each breath and the amount of times you breathe per minute. So these are the two factors that affect our ventilation rate. For example, the tidal volume can increase from 15% of the vital capacity to 50% during heavy exercise. So of course, during heavy exercise, we need to breathe in more and more oxygen, so the tidal volume increase by that time. So the frequency of inspiration shows similar increase. So there is a pattern that we need to dissect, and then if there is an interpretation question, so we'll use these data from the graph and use it to write it down on the paper. So let's talk about the control and regulation of breathing. To control the breathing rate, first of all, impulse from the ventilation center, that is again located in the medulla oblongata, travels along the sympathetic nerve. So this causes the intercostal muscles and diaphragm to contract during inhalation. Also, you can talk about the entire process of breathing in, inhalation, during inhalation. If the question is actually a 5 mark or 4 mark or 6 mark question, so then you talk about the entire process of the inhalation. If not, then this much is enough. Okay. As the lung inflate, stretch receptors in the bronchial valves. Stretch receptors here are located in the bronchial valves, send nerve impulses to the ventilation center. So then the more stretched these receptors are, the more rapidly they send the impulses. Eventually, these impulses inhibit ventilation center and it stops stimulating the breathing and muscles, leading to the relaxation of the muscles. This is called exhalation. So this is the process of breathing in and breathing out. So exhalation and inhalation. This is important. And lastly, to conclude, we talk about what is meant by the terms negative feedback and positive feedback, and the principle of negative feedback in maintaining systems within narrow limits. Okay. So, a simple definition of negative feedback means that a system that maintains a condition, such as the concentration of a substance within a narrow range. Receptors detect a change in condition, and as a result, effectors are stimulated to restore the equilibrium. So basically, let's look at the diagram or chart there in the left side. So, firstly, normal level of a particular factor. So suppose that our heart is beating in a very usual sort of rhythmic, natural rhythmic way, but then a receptor detects a change in a normal level of a particular factor. It detects any change, it could be any change in, suppose, your exercise, you're doing heavy exercise, so that could amount to a change and then more concentration more concentration of carbon dioxide increases, so the receptor just detects this change, and then the receptors then send signals, as we studied from the last previous slide. It sends signal to the cardiovascular center along sensory neurons. So this is nervous coordination, and then it sends signals via electrical impulses via the parasympathetic nerve. So that is the effector stage. So that to increase the level of heart rate, so that more oxygen can be pumped throughout the body. So then this action then returns back to the normal after a while when the carbon dioxide level decreases and pH increases. So that's when our body, or the state of the body, is returned back to normal. So that's the equilibrium. So after a few while, the body again notices a difference in the body, or a change into the change in the normal level, and then they use the same process to bring back the body in dynamic equilibrium. Okay, that's your negative feedback. And what is positive feedback? Positive feedback means a system in which effectors work to increase an effect that has triggered a response. So let's review that. Positive feedback means a system in which effectors work to increase an effect that has triggered a response. So here, for example, brain stimulates pituitary hormones, pituitary to release oxytocin, and this oxytocin makes uterus contract harder. So this pushes baby against cervix. Baby pushes against cervix, cervix stretches, stretch receptor in cervix and impulses to brain. So basically, over here, it's a case of hormone release from the pituitary gland in our brain. And the scenario describes when a female is contracting to push an unborn child out of her uterus and again through the cervix outside. So here the oxytocin makes the uterus walls to contract really hard so that the baby pushes out of the cervix. Baby pushes against the cervix as the cervix stretches, and the stretch receptors that is located in the cervix send impulses to the brain, and then the brain stimulates to do the same process again, and cycle repeats and repeats until the baby comes out. This over here, notice that over here in a positive feedback and in this scenario, the effect is heightened, or the effect is increased, so that the system is working to trigger a response. We have now reached the end of the chapter. Thank you so much for sticking so far, listening to my lectures, and I hope you found it useful. If you found it useful, please do let me know. And if you have any questions, feel free to ask me in the comment box below as well. Thank you.